So where did we leave off in the last video? We were talking about telomerase and how it counters the negative effects of DNA replication on the lengths of telomeres in eukaryotic chromosomes. So now we know telomerase is a very important enzyme, and we know the ability to lengthen the telomeres is critical for eukaryotic cells. And how do we know that? So this goes back to experiments in the 1960s with, I believe, they were fibroblast cells. So I think they may have been mouse cells, maybe they were human cells. I forget the details of this study. We're going to look at a different study in a moment. So what they found was when they isolated these fibroblast cells, these are not stem cells, they're not supposed to divide forever, but they were able to get these to grow and divide in culture. So we can take one fibroblast cell and we can use this cell to make a cell line. So what is a cell line? A cell line is a population of cells where every cell can be traced back to a single cell. So we can use this to make a cell line, and this one cell will double to make two cells, and these will double. So down here in this third generation we'll have 4 cells, and so on and so forth. We'll get 8 cells down here. And what scientists learned was after about 50 doublings, the fibroblast cells would stop dividing and the cell line would become senescent. I'm terrible at spelling this word. I don't know if that's right. Senescent. I might have to look that up. I'm going to go with it. So senescent. And what does that mean? Cells are dead, dying, and or not dividing, no longer dividing. So the cell line becomes senescent after about 50 doublings. And okay, let's pretend we don't know why. Maybe you don't know why, but you can probably guess based on the theme of this video and the last video of what's going on. You can probably guess what's happening in these cells. In the 1990s, scientists did an experiment, and in this case they are working with, similar to fibroblast cells. And I've never taught a cell biology course or development course or any course other than things related to genomics and bioinformatics and genetics. So my knowledge is rather limited when it comes to cell types, especially animal cell types. My focus is fungal genetics. So I know a relatively good amount about fungal biology, but not so much about different cell types. But these are going to be retinal pigment epithelial cells, and just like the 1960 studies, these are not stem cells, so they shouldn't divide forever. Now, but scientists can get them to divide and they can make cell lines from these types of cells. They can extract them from the animal and get them to divide in culture and make cell lines from them. So what they did was they took some of these cells and they made, I believe, 33 cell lines, with each line, and in each line they inserted a TERT dummy gene. Now essentially what this means is they just took a piece of DNA and they put it in each of these cell lines. And this DNA doesn't express a functional TERT enzyme. And remember, TERT is the telomerase reverse transcriptase component of telomerase. With telomerase, if you didn't see the last video, you might want to go back and watch that. Telomerase is made up of TERT and TERK. So they have 33 cell lines that have this TERT dummy gene, and they have 25 cell lines that have the TERT gene. They inserted the TERT gene into these cell lines. Now, okay, let's see what happens. I have their data right here, and I can't remember. I think I put this together a couple of years ago. Now, modified from this paper right here. So here's the reference down here, 1995, published in Science, one of the top science journals. Now, this is not in color, but all of these right here, you have to take my word for it. All of these should be in blue. If you look at the notes that are available in ReggieNet, these are all blue bars in this bar chart. And the TERT minus cell lines are blue. So we're calling any cell line that has the TERT dummy gene TERT minus. Now these should be red. All of these guys right here. So all of the red bars are the TERT plus cell lines. And so more specifically, what does this chart mean? So take a look at this bar. So these are all TERT minus cell lines, and what we're saying is that after 50 doublings, we can even call this the generation. At the 50th generation, we can see this little black circle right here. That means senescent. That means 1, 2, 3, 4, 5, 6, 7 out of the 33 TERT dummy gene cell lines are no longer dividing after 50 generations. So 51, let me see, 3 cell lines are no longer dividing after 51 generations. We have 6 right here. 6 of the 33 cell lines are no longer dividing after 52 generations. And so we can go way up here, and we have a couple of the TERT minus lines. Remember, all of these are blue. I really wish I had time to go home and print this out in color, but I need to get this video posted so I can go on to the next videos. So let's see. 1, 2, 3. These guys, there are 3 cell lines from the TERT minus of the TERT minus type that are still dividing at, let's say, 59, generation 60, and generation 63, but they're dividing less than 1 time per week. So it might be important to know that in very early generations, both the TERT minus and TERT plus cell lines were dividing at approximately the same rate. It's just when we get up here, the TERT minus cell lines senesce, or they slow down to dividing less than once per week. But if all of these right here, if it was in color, you could see that all of these are the TERT plus cell lines, and every one of them is still dividing at the time of publication. So at the time they took this data and published it, they had 1 TERT plus line that was in the 65th generation, and it was still dividing multiple times per week. They had 2 cell lines that were in the 66th generation, and they were still dividing multiple times per week. And they even had way up here, so they had 1 cell line that was in the 80th generation and was still dividing multiple times per week. So what does this mean? What does this mean? So essentially, it means the scientists took a mortal animal cell, the RPE cells, and they made it immortal by giving it a copy of the TERT gene. So telomerase requires two components, right? Requires TERT and requires TERK. So what this data suggests to me is that the reason the TERT dummy gene cells were all becoming senescent was because their telomeres are shrinking. And once the telomeres are gone, well, then the chromosomes ends aren't protected, and bad things happen to the cell. Cells lose genetic information, they're no longer healthy, they start dying, or they no longer divide. However, all of these up here can lengthen the telomeres because they have a TERT gene, a functional TERT gene. And so they can keep on dividing indefinitely. Now, notice the scientists didn't give the TERK gene to the cells. So human cells, RPE cells, all human cells, let's say from a wild type individual, have this genotype. So let's just focus on the RPE cells. And let's assume these RPE cells, if they were from a human and not from a mouse or a rat or a model organism. So with respect to the TERT gene, wild type would be TERT plus, TERT plus. And they would be TERK plus, TERK plus. So two wild type copies of both genes. Now, in normal cells that are not stem cells, that are not germline cells, the cells involved in making gametes, the TERT genes are off. So human cells have functional copies of the TERT gene, but they're off. They're silenced. And there's a reason, there's a hypothesis to explain why they're turned off and why TERT is only on in germline cells and stem line cells, cells that need to divide many, many, many times throughout our life. So the TERT gene is there, but it's off. It's not expressed. We're going to talk more about promoters and gene expression in the upcoming lecture. So this should be TERK, sorry. So the scientists were able to turn TERT on. They didn't turn it on. They gave the RPE cells, when they gave it a TERT gene, they gave it a TERT gene with an overexpression promoter. Essentially, they gave the cells a copy of the TERT gene that would be on and would express TERT at very high levels. So they gave the RPA cells a third copy of the TERT gene. The two natural alleles, right, TERT plus and TERT plus at the TERT gene locus on whatever chromosome it's on, they gave it another copy. And this copy is expressed. So that's why we're seeing an effect we're seeing this effect on cell lines that have the TERT plus gene, even though they already have two copies of TERT plus. Only the third copy, the extra copy, only that copy is on. But the scientists didn't give the cells TERK genes. So the genes that express the RNA. And my guess is that means that these are always on, whereas these genes are expressed in the RPE cell lines. It doesn't matter that these are expressed. They can't really do anything without expression of the TERT plus gene, right? Telomerase needs both components to function. Okay, so scientists were able to immortalize, turn mortal RPE cells into immortal cells by expressing the TERT gene. So, I think in lecture we watched a video from Stephen Colbert's show back, I think 8 years ago when he was on The Daily Show. He was interviewing a scientist who had made transgenic mice that had a copy of the TERT gene that was always on, and it seemed to reverse the aging process in these mice. Now, there is a reason why, or a hypothesis why most of our cells, unless they are cells that need to divide very often, like germline cells and stem cells, a reason why our cells keep telomerase, or the TERT gene, off in most of our types of cells. So, and it may be because of cancer. So let me explain. So if we can generalize cancer as the result of uncontrolled cell division. So, and cells possess many pathways that are supposed to regulate cell division and keep this from happening, keep cells from dividing in an out of control manner. And when a cell loses all of those regulatory mechanisms, it can start dividing out of control. Now, we can view telomere shortening as the cell's last natural defense against cancer, right? Because we see what happens when cells can't lengthen their telomeres, right? The cells die, they become senescent, they can't divide anymore. Probably because the telomeres are gone, the chromosome ends are no longer protected, and those chromosomes are destroyed or degraded. So a cell that becomes cancerous or precancerous and starts dividing uncontrollably, hopefully its telomeres will shorten to the point where the cell will die, because it will lose its genetic information needed to divide and grow. So we can view telomere shortening as the last natural defense our cells have against cancer. And evidence for this is seen in looking at analyzing cancer cells. So 100% of cancer cells, different cancer types, what's the way to say this? I hate saying 100%, right? It's the biology. But I'm going to say it anyways. 100% of cancer cells have found a way to lengthen the telomeres. So cells that divide out of control, the telomeres are going to be shrinking. Cancer, to exist, needs to find a way to lengthen those telomeres and counter the effect of shrinking telomeres due to replication. They found a way to do it. 80% of them, 80% do it by turning on telomerase. Maybe it's the TERT gene specifically, because our last analysis of this data here suggested that TERT is expressed at a level, even in cells where they don't normally have a functional telomerase gene. So 80% of cancer cells lengthen their telomeres by finding a way to turn on that telomerase gene. So some mutation that takes the telomerase or the TERT gene, moves it from the off state to the on state, so it's expressed. The other 20% use a different mechanism to lengthen the telomeres that we're not going to talk about, because I don't really know how that mechanism works. Okay, so that may be why our cells do not turn on, do not express telomerase in cells that don't need to divide throughout our lifetimes. But there are scientists and regular people who are very interested in the possibility that we could turn telomerase on in many of our cells where it's not naturally on, and that could reverse the aging process, and there seems to be data to suggest that. So that is really interesting for the future, is maybe we could dramatically increase human lifespan by learning how to turn on telomerase just enough that we don't increase rates of cancer, but we're able to extend human lifespan by maybe reversing the aging process by lengthening the telomeres in those cells of ours that do not naturally express the telomerase enzyme. Okay, so I think that's it for that one. In the next video, we are going to take a quick look at a disease and take a look at two new genetic concepts or terms that we haven't talked about yet. And this disease is also related to telomere shortening. Okay, see you in the next video.